Dear students, Assalamu alaikum and good morning. Today we will start the new chapter. That is, chapter number 4, Cells and Tissues. Learning objectives. At the end of the lesson, dear students, you will be able to: Number 1. Understand light microscope and electron microscope. Number 2. Learn about the types of electron microscope. And lastly, know the differences between light microscope and electron microscope. This chapter is about cells and tissues. All organisms are composed of cells. Some are made of single cell and are called as unicellular. Others like us are made of many cells and are called as multicellular. Tissues are group of cells in multicellular organisms. This chapter deals with cells, their internal structures and tissues. First topic is microscopy. First, see its definition. The use of microscope is known as microscopy. The first compound microscope was developed by Zacharias Janssen, in Holland in 1595. It was simple, a tube with lenses at each end. Its magnification ranged from 3x to 9x. This means that it was able to enlarge the object 3 to 9 times its original size. On this slide you can see the first microscope made by Zacharias Janssen in Holland in 1595. Here are important terms which are used in microscopy. We will discuss two terms. First one is magnification. It is the increase in the apparent or true size of an object. It simply means the degree to which something is or can be increased as compared to its original size. Second term to discuss is resolving power or resolution. It is the measure of the clarity of an image. It is the minimum distance at which two objects can be seen as separate objects. Human naked eye can differentiate between 2 points, which are at least 0.1 millimeters apart. This is the resolution of human eye. This means if two objects are 0.05 millimeters apart, human eye would not be able to differentiate them as two separate objects. Magnification and resolution can be increased with the help of lenses. There are two types of microscope: light microscope and electron microscope. First to discuss in detail is light microscopy. Its main principle of working is a light microscope works by passing visible light through a specimen. It uses two glass lenses. One lens produces an enlarged image of the specimen. Second lens magnifies the image and projects it into viewer's eye or onto photographic film. A photograph taken by a microscope is called a micrograph. This is diagram of light microscope. See different parts carefully. We will use this microscope in laboratory for certain practicals. As concerns the magnification, a light microscope can magnify objects only about 1,500 times without causing blurriness. So, its magnification is 1500x. If you see the resolution, its resolution power is 0.2 micrometer. Light microscope is not advanced type. It has the drawback that it cannot resolve the objects smaller than 0.2 micrometers, which is the size of smallest bacterium. Also, it cannot show the details of internal structures of a cell. Different types of bacteria seen under light microscope are shown in this diagram. We can see bacteria but cannot see internal details of cells. Second type of microscope is electron microscopy. It is the most advanced type of microscope. Its main principle of working is in M. Object and lens are placed in a vacuum chamber and a beam of electrons is passed through object. Electrons pass through or are reflected from object and make an image. Electromagnetic lenses enlarge and focus the image onto a screen or photographic film. This is the diagram of electron microscope. You can see it is very complex and large and needs a lot of space for installation. Only experts can handle it. Now see magnification of this microscope. Electron microscope can magnify objects about 250,000 times than their original size. Resolution. It has much higher resolution than light microscope, which is 0.2 nanometer nm. We have discussed both types of microscopes. Now we will study advantages and disadvantages of electron microscope over light microscope. First, advantages. First one is that electron microscope has resolution power which is thousands times more than light microscope. Second is that electron microscope can detect individual atoms, cells, organelles and even molecules like DNA and protein. Now see disadvantage of electron microscope. Electron microscope cannot be used to study life processes because specimen must be held in a vacuum chamber which does not contain air, so living things cannot survive. To study living processes like movement of amoeba under microscope, a light microscope is a better option. In this view you can see micrograph of human blood taken by light microscope on the left side and by electron microscope on right side. It is so clear that electron microscope is showing clearly human blood cells as compared to light microscope. Even platelets are visible in the micrograph shown by electron microscope. Types of electron microscope. Biologists use two types of electron microscopes. First type is transmission electron microscope, TEM. In TEM, electrons are transmitted, means pass through specimen. It is used to study the internal cell structures. For example, study of mitochondria, nucleus, ribosomes etc. This is the diagram of transmission electron microscope, which is very complex and large. These are images taken by transmission electron microscope. On right side there is image of Golgi bodies and on left side there is image of cells showing internal structures. Second type of electron microscope is scanning electron microscope, SEM. In SEM, electrons are reflected, meaning thrown back from the metal-coated surfaces. It is used to study the structure of cell surfaces. For example, SEM is used to learn much more about bacteria and viruses. This is the diagram of scanning electron microscope. This is also complex like other type. A scanning electron microscope shows the images of cell surfaces. You can see surface of head of mosquito showing eyes on right side and surfaces of pollen grain on left side. Dear students, note down the homework now. You have to complete the given worksheet. Have a good day. Stay blessed and safe. Thank you.